We've gone through carbohydrates, we've gone through proteins. It's time to move on to lipids, the digestion of lipids. But there's something unique about the digestion of lipids. They go through kind of like a pre-treatment before the enzymes start to work, or at least before the enzymes work efficiently. That's where bile comes into play. The bile salts will aid with emulsification of the lipids. What that means is you're taking this large chunk of lipids and breaking it up into smaller lipids. The entire purpose is meant to increase surface area. By increasing surface area, you allow the enzymes to be more efficient. The enzyme we're looking at is pancreatic lipase. So that means pancreatic lipase goes from the pancreas into the duodenum. So for lipids, chemical digestion initiates in the duodenum. Mechanical digestion, though, that initiated in the mouth when you're chewing. All right, so absorption now. This gets a little more interesting because lipids are fats. And you know that your blood is mostly water-based. Water and fat doesn't really mix very well. So what we start with is the breakdown from the enzymatic activity, so from that pancreatic lipase, is glycerol and short-chain fatty acids. So just part of the breakdown process. They're going to be absorbed right into the blood capillaries from the villi, transported to the system, and up through the hepatic portal vein. Pretty much just like the previous two we talked about, the proteins, amino acids, and the carbohydrates, the monosaccharides. But things get a little different after that. Now, if you're absorbing the monoglycerides or the rest of the fatty acids, not the short chain, now things change. What ends up happening is the bile salts will kind of cluster with them, cluster around, and you end up with lecithin forming micelles. These micelles are kind of like a coating around the actual monoglycerides, around the fatty acids. Now you can have these released into the micelles for epithelial cells. Now once you go with the epithelial cells, that actual monoglyceride, that actual fatty acid chain, that then forms chylomicrons. That's because there'll be proteins that'll coat and cover them. Again, they can't function very well in a water-based environment. That's our cells, that's our blood. Those chylomicrons then go into the lacteals. Remember, the lacteals are part of the lymphatic system. They're not the same makeup as blood. So yes, eventually we'll get into the blood, but at least for now, we're starting in the lacteals. And they'll be transported up until they eventually are dumped into the circulatory system. So to kind of take a look at what's happening here, you're starting with a big fat globule up top. That needs to be broken down. So the initial part is the bile salts going through emulsification. By emulsifying, you still have lipids or fats. They haven't changed. They're just smaller chunks. By doing this, you've increased the surface area, given more surface area for the enzymes to work. Now those enzymes are going to digest as much of the lipid as possible. What ends up happening then is, you notice right here, we have these micelles. They're coated because they can't simply be absorbed. They're coated and they will get into the cells still. They still are sent in through that simple columnar cell. But once in there, they then are forming into chylomicrons. Those are going into the lacteal. So the lacteal will then transport them out and away from that location. So we have a little different process for some of the breakdown components of your lipids. So the lipids themselves, not a lot of different chemicals coming in, but the process of how to package them and how to transport them is different. Because you literally have bile from the liver. Sure, it can go through the gallbladder and then come in, but it enters the small intestines. That's emulsification. You then have the pancreatic lipase coming out of the small intestine. That's the actual enzyme that'll break it down. You're breaking it down either to glycerol and fatty acids or monoglycerides and fatty acids. Depending on which grouping you're in depends on how you're absorbed. Are you going directly into the capillaries, glycerol, or are you being packaged into micelles and then chylomicrons, the monoglycerides and the larger fatty acids? So the packaging, the transport, is what's different in the lipid digestion. And our fourth of the macromolecules. We've gone through carbohydrates, proteins. We just went through lipids. Now we're left with nucleic acids. Nucleic acids are going to have the ribonuclease and the deoxyribonuclease. Or ribonuclease for RNA, deoxyribonuclease for DNA. And they're both being produced from the pancreas, which means chemical digestion of your nucleic acids starts in the small intestines. Mechanical digestion, that can potentially start up in the mouth from chewing again. So that means we have 4 different molecules now. We have the carbohydrates, their chemical digestion initiated up in the mouth. We have the proteins, their chemical digestion initiated in the stomach. The lipids we just talked about, and now the nucleic acids, both have their chemical digestion initiating in the small intestines. Absorption, right back to that active transport again. You need energy to absorb these components. And just like the other ones, for at least the majority of it, not every piece of lipid, but we're heading up the hepatic portal into the liver. Liver processes it and then sends it on its way. So again, we're looking at liver, ribonuclease, deoxyribonuclease, but we also have some brush border enzymes that can help out in the process. Again, the liver is the main one, but you do have some helping aid from the brush border enzymes. All right, the 4 macromolecules are set now, looking at the physiology of digestion of each one of the 4 macromolecules.